Hi everyone, so we're about to start off on chapter 3 where we're talking about macromolecules. And so for our devotional for this chapter, I wanted to talk a little bit about discipline. So sometimes it's fairly easy to think about a devotional that fits with the topic, other times a little bit harder. But this one I think is a little bit better because we're at a point still at the beginning of the semester, but where you're starting to see what the semester is going to look like. And so a little bit of talking about discipline. So discipline yourself for the purpose of godliness, for bodily discipline is only of little profit, but godliness is profitable for all things. So whatever you do, kind of keep in mind that higher purpose. And also start to think about what areas in life you might be lacking in discipline. I know this COVID pandemic has really kind of started pointing out to me, unfortunately, some of the things that I am probably lacking discipline in. And so I have a lot of things myself to work on in that regard. But especially with regards to studying and kind of getting off on the right foot, just make sure you're being disciplined and in any other thing as well. But above all else, keep that godliness aspect, the better good, in mind. All right, so let's talk about macromolecules, so biology. So carbon is the building block of all biological beings, and there's a reason for this, because it has that tetrahedral structure. So we talked about molecular shapes, and carbon, because its valence is 4, makes 4 bonds. So it can not only bond with a lot of different things, but it starts to create those shapes. So it also binds to nitrogen, hydrogen, oxygen, sulfur, and in doing so there's a lot of shapes, and we end up with our organic molecules, like this one is cholesterol. So you can see here that the carbon building block bound to, in this case, mostly hydrogens. This is mostly a hydrophobic hydrocarbon, but also oxygen in this case as well. But this creates an almost infinite amount of variety of carbon molecules that can be made. Carbon bonded molecules are also very strong. So think about what the strongest thing you can think of. Well, diamonds are extremely strong, and diamonds are just tightly packed carbon covalently bonded to other carbons. It's a carbon-carbon molecule. So not only do we have variety with carbon as the building block, we also have strength. So then linked onto those carbon building block molecules, we have what's called functional groups. And these functional groups are called that because they actually have functions. They can react. They have a lot more properties of interaction with other molecules, of reactions for enzymes, all kinds of different things. So at this point, you just need to recognize these functional groups. So these are in table 3.1. I'm not going to spend much time going over this because this is something that you can basically start to memorize and recognize on your own. But we will be talking about these and how they function in biology throughout the rest of the course. So we have our methyl group, just carbon with hydrogens, our amine group, nitrogen, not hydrogens, phosphate groups, these are going to be really important for nucleic acids, for example, hydroxyls, alcohols, OH, carboxyl groups, C double bond O, OH, this is important for nucleic acids, carbonyls, sulfonyl hydryls. So basically study table 3.1 and really get to know that pretty well. All right, the next thing we need to talk about for macromolecules is polymerization. So the whole idea of polymerization is that we're taking a basic building block, a monomer, and we're linking it together to make these really long polymers. So the monomer for this example would just be A. So A might indicate anything. A might indicate an amino acid, sugar, or whatever, but this is a single molecule, right? And then when we start to link them together, A bound to another A, and all these molecules together make that polymer. And this happens in biology through a process called a dehydration reaction. And I'm going to go over these in the next video in a little more detail. But a dehydration reaction, hydration, water, just as when two monomers react, the OH group, the hydroxyl group, and the hydrogen group basically are lost, hence the dehydration, and that creates a bond between those two monomers. And then this keeps happening and we get polymerization. And so the dehydration means a molecule of water is removed each time, and again this just repeats as we polymerize our monomers together. Well, if you can build a polymer by dehydration, you can break it down by hydrolysis. Hydro, water, lysis, breaking down. So those bonds were made when a water was lost. If water comes in and it can break that bond, leaving you the one monomer out here back with its OH group, and the new end, with a hydrogen. And so hydrolysis can continuously come in and break down each of those polymers, break apart those monomers sequentially. All right, and so the building blocks that we're worried about in biology are sugars. All right, and sugars linked together create the polymer, polysaccharides, and carbohydrates. Nucleotides, that's the monomer of nucleic acids like DNA and RNA. Amino acids, that's the monomer that builds together and creates polypeptides or proteins. And so these are the monomers that you need to understand. We're going to go over them in detail. And these are the polymers that they create. And so you really need to understand and have these ideas linked together in that regard. And then the last one, a little more simple, but fatty acids are the monomer that build together to make fats and basic lipid membranes like phospholipids and so forth. All right, so the main goals of this chapter, or at least the first part of it, is understand the structures and roles of carbohydrates, nucleic acids, proteins, and lipids. So what do they do? What's their functions? And how do they link together? Know how to number the carbons in a sugar, understand the properties of saturated versus unsaturated fatty acids, identify hydrogen bonds between nucleotide bases. So we're going to get into this with DNA and why the base pairing works. And then protein folding, we'll spend at least a full day, if not a couple of days on, talking about protein folding and structure. So that's where we're going to get to. This first part here will be on Friday and in the next video that you're supposed to watch ahead of time as well. All right, thanks.